About how to determine onion epidermis. First of all, I would like to tell you about the onion. Onion locally, scientifically, it's Alieum cepa. Locally, it's called pyaz. So, okay. Now we are going to determine its epidermis. इसको हम प्याज कहते हैं और इसकी आज हम एपिडर्मिस निकाल के ये जो इंस्ट्रूमेंट है जिसको माइक्रोस्कोप कहते हैं इसके अंदर देखेंगे, इसके सेल्स को हम वेरीफाई करेंगे कि ऑनियन एपिडर्मिस के सेल्स कैसे दिखाई देते हैं. क्लियर? सो फर्स्ट ऑफ ऑल we will have introduction about apparatus. So that is light microscope. We will use a plain slide and then we will use a cover slip. Cover slip is basically a thin glass apparatus used to cover your specimen and clarify epidermis of the cells in detail. Apart from this, so it's very thin, having thickness less than a paper. So that will be used in experiment. Then we will use a dissection box. Dissection box is basically used for the dissection of frogs. So here we will use razor and forceps and camel hair brush in order to move our specimen from this petri dish to the slide. So we will use the dissection box, petri dish with water and beaker. Ink दे दो, ये अब निकले. You can see a blue color solution. This solution is called as methylene blue solution. So let's start our practical. We will use another instrument that is called as razor. That is used basically for cutting. So we will have a cut on the surface of this onion. Keep staying here. Okay, you can see there is a thin layer inside this cutting. We are going to move this thin layer. This thin layer is epidermis of onion. We will shift it into the petri dish in the water in order to refresh all the cells to be clearly visualized under microscope. So be careful in removing epidermis and shifting it into water. Try to take the smallest piece as it will make epidermis cell more clear as compared to the cells in the folding and in the layers. So if we use the cells in the folding and layers, it will not make sure that the onion epidermis is visible to you. Be careful in removing this epidermis from onion. Make sure don't use your hand to rupture this layer. It's a thin layer like watery layer. So pour it into a petri dish having water, and then very carefully you have to first pour a drop of water here on your glass slide. Place a drop of water on the glass slide. Then very carefully try to cut a very small piece of this onion epidermis. Very small piece of this epidermis. While placing it in water, make sure you are not rupturing the cells of epidermis. You have to shift this onion epidermis on glass slide like this. Is it visible to you?
Yes.
Okay, then adjust roughly your piece of onion epidermis into pre-settled light microscope. So here we will see the cells of, after settling your piece of onion epidermis on glass slide, you have to pour a colored solution in the form of methylene blue with the help of dropper. You will have to drop only a drop of water on your specimen. Then again visualize your piece of epidermis in microscope. After you can see that I can see the cells properly in rectangular form. Then you will have to settle your glass slip, cover slip. Cover slip should be settled in a way that there will be no air bubble in epidermis and cover slip. If there is presence of any air bubble, then that will be having negative marks for your practical. Slightly taper if in case you are observing some of air bubbles. You can taper your slide like this. That will remove your air bubbles.